Thank you so much. This is the second or third time I've had the privilege of speaking to you from this very podium, and I thank the meeting organizers for yet another opportunity. So I'm going to talk to you a little bit about T-cell lymphoma, and some of what I'm going to say has the potential to be practice influencing. But a lot of what I'm going to say, I hope, is going to be more rethinking the paradigm and paradigm changing. Because we'll talk a little bit about the unfulfilled promise of chemotherapy in T-cell lymphoma, and I'm going to try and make the case that what we've been doing for the last 20, 25 years or so, being somewhat addicted to CHOP-based chemotherapy, may be somewhat flawed if we look at that historical record and the evidence in support of it. We'll talk a little bit about the drugs that we have at our disposal, but unfortunately, some of those drugs are under attack, at least in the United States, where romidepsin, widely regarded as a very good drug for the disease, was recently pulled from the market for patients with peripheral T-cell lymphoma, really diminishing what we have at our disposal for these patients. I'm going to talk a little bit more about novel combination platforms. That's the work that I do in my laboratory, and we translate that to the clinic. And I'm going to try and share with you that maybe how we think about this disease with regard to our addiction to chemotherapy versus our stimulating our innovative instincts to develop new regimens that are not necessarily predicated on chemotherapy may have its merits. And so I start with this famous paper from our good friend Rich Fisher and colleagues, published back in 1993. And you all know this paper because it's the paper that compared CHOP against other dose-intense regimens, MACOP-B and Embicod and Promacytobam. And the title of the manuscript, as you can see, is Advanced Aggressive Lymphoma, non-Hodgkin's lymphoma. In reality, this became the standard of care largely for all B-cell lymphomas and by extension T-cell lymphomas. And what you see from the paper is the well-known conclusion that everybody knows that other dose-intense regimens were no better than standard CHOP-based chemotherapy. But I'll argue the point here that the classification scheme used back then for this study was actually the working formulation. And how many people, I'm just curious, besides all the old guys, how many people remember the working formulation? Could anybody here cite the working formulation?
Only you.
Only me, that's right, because I'm one of the oldest. And so that's probably almost right. And the working formulation back then used this classification of thinking about the clinical behavior of lymphoma, low grade, intermediate grade, and high grade. I'm going to ask another question. How many people, and you've all read this paper, I'm sure, Mubarak, had you read this paper 100 times in various settings as fellows. How many of you actually knew that the national high priority study that established CHOP as a standard of care used the working formulation? And the working formulation had its limitations, and if you look back at the paper, you see patients with all these different subtypes of lymphoma were classified as aggressive. So they were included in the study. And that would include today people that had Burkitt's, aggressive mantle cell, transformed lymphoma, large cell lymphoma, and peripheral T-cell lymphoma. And I beg the question, where's T-cell lymphoma? I've read this paper at least 25 times. I cannot find the word T in the paper except in the use of the word the. That's it. There is no discussion of peripheral T-cell lymphoma. But for some reason, we as a community said, this is our standard of care. And I'll challenge anybody to say, is it possible that a methotrexate-containing regimen isn't better in this setting? It's possible. And I will argue that we've spent a lot of time, 20 years, taking CHOP-based chemotherapy and using it in T-cell lymphoma, and then we argue chemo's not very good. I'll argue maybe chemo has some value, but maybe we're not using the right one. And the greatest innovation we've been able to muster in this disease for 20 years has been to add drugs to CHOP-based chemotherapy. And I'll talk about two, brentuximab and romidepsin. Arguably the two best drugs people think of when they think about T-cell lymphoma. And we've taken this historical paradigm and just added new drugs to CHOP, thinking, oh, let me add one good drug to 4 bad ones and I'm going to make a great 5-drug regimen. So that's what we've been doing. So when you say it like that, everybody scratches their head. And in fact, Echelon 2, which uses brentuximab, we heard a little bit about brentuximab, and in this particular case, brentuximab, was studied in patients with peripheral T-cell lymphoma. But in Echelon 2, more than 2/3 of the population actually had anaplastic large cell lymphoma. Happens to be the subtype that is known for CD30 expression and the highest single-agent response rate to brentuximab. And the company notes that that was done to satisfy the European regulatory agencies. And in that study, there was clearly an advantage on the entire population for those that got brentuximab. But what's clear is if you look at this data and you look at the breakdown, and this has been one of the things that has come out really interestingly, is that when you look at all the other PTCL that are not anaplastic large cell lymphoma, only anaplastic large cell lymphoma benefited. Patients with other peripheral T-cell lymphomas did not benefit from Echelon 2 based therapy, did not. Now we know that the expression of CD30 in the non-anaplastic large cell lymphomas is much more diverse. So the trial had a requirement that you had at least 10% expression of CD30. When the FDA saw the data, the US FDA said, well, it's pretty hard to quantitate CD30, so as long as you're CD30 positive, irrespective of quantity, they'll approve the study. So Echelon 2 is approved for all patients with PTCL, even though the statistical benefit of the regimen is in patients with only anaplastic large cell lymphoma. And so for those who like to challenge me and say, well, Owen, chemotherapy isn't all that bad in T-cell lymphoma. Well, I like to point to this great data from the British Columbia Cancer Agency up in Vancouver. And what they did is they went back and they looked at all patients with T-cell lymphoma and asked the question, what is your second progression-free survival and your second overall survival? You know that if you have relapsed anything, your prognosis is better in first relapse than it is in third or fourth, right? So this would be the best outcome you could possibly expect for any patient with relapsed lymphoma because they're looking at you in your first relapse, not your third or fourth or fifth, which is where we study all these drugs. And so in fact, the second progression-free survival is 3.7 months. The overall survival from your first relapse, 6 months. This now makes T-cell lymphoma worse than acute myeloid leukemia. It's amazing. And in this setting here, what are we using to try to salvage these patients in relapse? It's chemotherapy. It's all chemotherapy. So if chemotherapy is so great, then why is these data so bad? And in fact, if I sort of look at the evolution of what we've been trying to do in T-cell lymphoma over 2 decades, we got the famous Fisher paper. You see a cluster of studies where a lot of people said, oh, let's add Campath, let's add Vorinostat, let's add Denileukin-Diftitox, let's add everolimus to CHOP chemotherapy. All of these studies, negative studies. And in fact, arguably the 2 best drugs we have in the disease, brentuximab and romidepsin, also negative studies, except in the case of Echelon 2 in ALCL. So our evolution in terms of what we're doing in T-cell lymphoma, we're not evolving. We're in stagnation. And so I'll argue that this line is somewhat contrived. I drew this line. It's got more of a slope than I think it actually deserves. And look at the ROCHOP data. Everybody, I was involved in this study from the moment it became an idea. I was involved in romidepsin when it was still FK whatever. And here's the romidepsin study. God rest his soul, Bertrand Coiffier spent 3 years of his life doing the phase 1 for the ROCHOP study. 3 years. I was at meeting after meeting after meeting where he said, we don't have the final dose yet. It's very toxic adding another drug. But what oncologist in this room doesn't know that adding a 5th drug to a 4-drug regimen is a problem because you're going to get toxicity. And in fact, they did it, no difference, none whatsoever in adding romidepsin. So if we take our 2 very best drugs, brentuximab and romidepsin, and we add them to CHOP-based chemotherapy and they both fail, should we be rethinking the paradigm? And I'll argue we need to rethink the paradigm. And we're so stuck in our ways. Guess what the follow-on study is for this? Anybody know?
6th drug.
The 6th drug, exactly. CHOEP. They want to add romidepsin to CHOEP. Oh, because we forgot the etoposide. That's why it didn't work, right? That's how creative we're getting in the field. And I'll argue we have to change ways. And this would be how I think about the field today. We've done this for a long time, right? It has a lot of advantages. It can be fast and patients get a known regimen. But there's a lot of disadvantages. The logic of adding one drug to 4, one drug to 5, I don't know. I don't know what it is. It's often very toxic, and we've had two decades of negative studies. But if we think about using some of the novel drugs that we have, and every time I give this talk, somebody always says, well, we need new drugs in T-cell lymphoma. Yes, we need new drugs for every disease we're fighting. But there may be advantages here because a lot of the new drugs we'll talk about are lineage specific. They only work. Pralatrexate, romidepsin, brentuximab, belinostat, chitomide. They only work in T-cell lymphoma. And I'll argue, you know what? We got some good drugs at our disposal. We're just not using them very well. And so there are some advantages. Those drugs may respect biology. They build on positive findings. There's some disadvantages because we're kind of starting from the beginning. But what I want to talk about is this as a potential paradigm-changing opportunity for us in T-cell lymphoma, and to try to resist the seduction that comes with just adding yet a 4th or 5th drug to a regimen we don't think works real well. And so this is just our effort from a paper that Dr. Markey and I published years ago. And admittedly, there are no home runs here. We got drugs, but there's nothing that's like knocking us out of the park. These are FDA registration studies with Romi, Belino, Prala. They're 30%, 25, 30% drugs. And so yes, we need new drugs, but if you look at what our friends in multiple myeloma have done, they've totally changed the paradigm. Gone are the days of melphalan, in are the days of proteasome inhibitors and IMiDs and daratumumab, right? They've taken the new drugs they had. Steroid, for God's sake, was the most active drug they had. They've changed the natural history of the disease by using what they've got. And in fact, when we start looking at combinations of these drugs, look what happens. We've taken Aza-Romi, my group, and Prala-Romi, based all on lab work. I'm not going to go into too much lab work, showing you 80, 90%. Somebody said we couldn't cure T-cell lymphoma without chemotherapy. I agree. But maybe we can cure them with these drugs. I've treated a lot of patients with Aza-Romi. I have 5 patients with angioimmunoblastic T-cell lymphoma that got azacitidine romidepsin in their 5th year after failing everything under the sun, disease-free. Are they cured? I resist using the word, but it's certainly suggestive. And so I think there may be settings here where we can build on what we've got to try to create something better. And in fact, where I like to always start in thinking about how to develop a new drug regimen for a disease is with biology. Because if we're just picking chemotherapy out of the air and throwing it against the wall, we're just hoping for something good to happen with something that's potentially noxious. How do we target underlying biology? So the classifications, and many of you have seen this from Iqbal, the gene expression array can help us in classification. But what's the problem with this gene expression array data to date? The problem is it has not given us any hard clues about biology to go after. I can make the case pretty reasonably. The biology at play that we need to target in T-cell lymphomas is epigenetic biology. Unfortunately, epigenetic biology doesn't pop up in any gene expression array. And so how do we now merge our understandings of the molecular biology via gene expression array with other complex pieces of biology, like understanding the epigenome? So my lab years ago, we do a lot of high-throughput chemical screening and high-throughput genomic screening, and we asked the question back then, show me all the drugs on the planet that synergize with romidepsin. And remarkably, guess what 2 drugs popped up? We screened thousands of drugs, pralatrexate and azacitidine. So you might look back and say, well, Owen, you didn't need to spend $50,000 doing high-throughput chemical screening. I could have told you those 2 drugs would work. Well, we did it in an agnostic way, asking the question. Let the data drive us what we do. And in fact, here's the data we did with Prala, and I won't go through all the detail, but this is looking at Prala plus Romi at half the maximum tolerated dose. And herein lies a value of what I'm talking about. If we identify synergistic drug-drug combinations, what is the clinical benefit of synergy? I get to use the IC10 or the IC20 of a drug in combination, but be able to magnify the response rate. In theory, rather than adding a 4th or 5th or 6th drug to CHOP at the maximum tolerating dose and then discovering, oops, I get a lot of toxicity. What if I took a pharmacologic principle like synergy and said, well, let me use drugs potently synergistic, so this way I get to use lower concentrations of them, avoid the toxicity seen in combination while magnifying the benefit. That's the platform. And lo and behold, that's exactly what happened. MTD of Romi, MTD of Prala, the combination produces a survival advantage. We take it to the clinic. And when we did the synergy analysis, guess what? We only did it in T-cell lymphoma. I was not interested in developing a drug regimen for large cell or follicular. So we only did it in T-cell lymphoma, and we asked the question, show me the combinations only synergistic in T-cell. And so that's exactly what we got. The overall response rate, 57%. In B-cell lymphoma, 30%. In T-cell lymphoma, 70%. So from the very beginning, from its inception, we wanted to start out finding drugs active in T-cells, set the platform, and in fact, that's what we did. Now, the other combination we looked at, azacitidine. There's a lot of preclinical data, and I'm happy to answer questions. But we did the exact same thing with azacitidine. And some of you are thinking, that's not so clever, Owen. They're both epigenetic drugs. You made the case it's an epigenetic disease. Yes, but it's good to have the data. But look at this. A response rate in T-cell lymphoma, 73%, B-cell lymphoma, 10%. Has anybody ever seen a drug regimen have a better response rate in T-cell lymphoma than B-cell lymphoma? This is the first, right? This is very, very unusual. And in fact, this is experience from the phase 1. And in the phase 2, we treated patients who were treatment naive. I won't have time to go into the data, but we did study, and there's a very good paper from Dr. Ma from my group where she did a historical retrospective survey of over 250 patients we treated with T-cell lymphoma with chemo and new drugs. And she asked the question, show me, and I say this because of the great transplant talk that we heard a few moments ago. We said, ICE, I'm not so impressed with ICE in T-cell lymphoma. In fact, all the data says it ain't so great. So she compared 10 patients we had with that got AZA and Prala-Romi prior to auto stem cell transplant with like 40 or 50 patients that got standard ICE chemotherapy. Of the 10 patients that got Prala-Romi or AZA-Romi prior to auto versus the 50 that got ICE, 30% or 25% of the patients that got ICE were alive at 2 years. 100% of the patients that got AZA-Romi or Prala-Romi were alive at 5 years. Does that suggest that maybe we should be rethinking this paradigm? And rather than pushing the chemotherapy option, begin to get creative and ask, well, what happens if we treat treatment-naive patients? 70%. So we see pretty good response rates. What's real interesting is this real high signal of activity in the T follicular helper lymphomas like angioimmunoblastic and TFH. And there's a real reason to think why these could be sensitive, but this continues to emerge as a subtype that's very sensitive to the combination. And in fact, this just shares the swimmer's plot. You can see some pretty long responses. We got a bunch of patients out 5 years following this regimen, and a very favorable waterfall plot with a lot of patients in CR. But this is the curve I love to show the most. Because has anybody seen a curve where the progression-free survival with any regimen in T-cell lymphoma is better than the progression-free survival in B-cell? And in fact, we haven't. And so this is the progression-free survival for T-cell lymphoma. It's about 9 to 10 months versus B-cell lymphoma. And one of the great limitations of developing drugs in T-cell lymphoma has always been the progression-free survival of Prala, Belino, Romi, Chitamide, and brentuximab is only like 2 months, 3 months. That's like the progression-free survival I just showed you from the paper from Mac, which was 3 months. One could reasonably argue we're not doing anything to improve the progression-free survival of these patients. And so changing the natural history of the disease by changing this progression-free survival interval, I think is key. And it's something we need to evaluate prospectively. Look at the overall survival curve here for patients that were treated on the phase 1. And again, this is the same story from the phase 2, reinforcing everything else I just showed you. And so this data has been received well by the US FDA. They've actually asked us to do a randomized study, so we got a huge grant from the FDA where we're doing a randomized study of azacitidine using oral plus Romi versus investigator's choice. This study is now ongoing, and if positive, could begin to pave the way for how we think about developing new combinations. And we've already built on this. Dr. Markey will be presenting at ASH the patients with oral AZA, romidepsin, and Prala-Romi getting checkpoint blockade, pembrolizumab. And in that study, there's a real logic to add checkpoint inhibitors, and in those studies, the response rates are pushing 100%. So what else is on the horizon in T-cell? And I have to tell you, the horizon looks pretty blurry to me, but there's not a lot of new drugs out there. But there is one, and I want to use this as an example to underscore the point I just made about combinations. This is a drug called Tolinapant. It's been studied in PTCL and CTCL, as I'll show you. It's a very interesting drug. It started out as an inhibitor of apoptosis, a CIAP inhibitor. But it turns out it has lots of immunologic effects inducing immune response against the tumor. The biology is fascinating, the pharmacology super complex. But Dr. Zanzani and colleagues presented an update on Tolinapant. We were very much involved in this study, looking at it in patients with PTCL and CTCL. And this is really your classic single agent drug, another new drug in T-cell lymphoma, looking like all the others, right? You look at this and you say, oh hum, right? It's got a response rate of maybe 20 to 25%. Here's the swimmer's plot. Okay, Owen, it looks just like a lot of the other drugs. And this drug is promising because I've treated patients with PTCL that are in remission 3-4 years later on this drug. So there are these really phenomenal responders, but it's certainly not everybody. So in the laboratory, what did we do? We screened Tolinapant against all our different cell lines and found some cell lines are incredibly resistant, T-cell lymphoma. Some cell lines are very, very sensitive. And then we asked that question I shared with you before. Show me drugs that synergize with Tolinapant in T-cell lymphoma. And what popped up? What popped up was decitabine and azacitidine. Now, for some reason, I think there's an emerging theme here. You know, the French studied azacitidine. They're going to report data at ASH that's only so-so on the single agent. But the synergy that we've seen with Tolinapant and decitabine is off the roof. It's off the roof. And it turns out that there's an oncogene, we're going to call it, it's called RIP kinase 3. And RIP kinase 3 plays a very important role in governing the sensitivity to Tolinapant. So if I go back and I take these resistant cell lines and I incubate them with Tolinapant and decitabine, they're all now super sensitive. Because it turns out RIP kinase gets hypermethylated. And by using a hypomethylating agent, I now can render all these T-cell lymphomas super sensitive to the drug by looking at the combination. And so now we're about to begin a national study looking at the combination of Tolinapant with hypomethylating agents. And for me, it simply makes the case, we got to stop thinking about adding stuff to chemotherapy. People are adding all sorts of things to ICE and T-cell lymphoma, but we should be thinking about how do we replenish the drugs that are getting pulled from the market underneath our noses? And how do we think about these clues coming along, like Tolinapant? How do we make them better, like our friends in multiple myeloma have done? And this is the unfortunate history of romidepsin, a pretty good drug. 25% of people respond, but if you respond, your response can last way over a year. That makes it a pretty interesting drug. But because of that phase 4 study that I showed you where they added romidepsin to CHOP and it was negative, the FDA says, no, you're no good, off you go. And what's the problem? The problem is my colleagues said that was the best study to do to show unequivocally the benefit of romidepsin. And I was at all those meetings saying, no, I wouldn't do that. I would get creative. But we did it. And guess what happened? The FDA said, that was your choice. You did it. Now you pay the price. And the price is the price the patients will pay because now we don't have the drug. And so in my lab, I'm a chemist by training. And so I love chemistry, and wherever I could talk about chemistry, I like to. We went back and we said, well, the heck with this. We're going to go back and we're going to create a new portfolio of drugs in T-cell lymphoma. We're going to start with romidepsin because almost every drug I combine with romidepsin in T-cell lymphoma, I get profound synergy. So we made a nanotherapeutic polymer of romidepsin. And this is all the data. It's as good or way better than what we see with naked romidepsin. And instead of giving romidepsin, how many people here have given romidepsin? Is it approved here? You don't have it? So it's a very popular drug in the United States and Europe. It's a 4-hour infusion once a week for 3 weeks in a row. And it's got its issues with the infusion. These kinds of formulations of the drug, we can give over 30 minutes once a month or so. So we're now using these polymers And embedding in these polymers, we're putting all sorts of other drugs like azacitidine, like tolinapant, like other drugs. And so within these complex chemical nanoparticles, I can create new combinatorial therapies that we're starting to create now for patients with T-cell lymphoma. And so that's just a flavor of what I think is coming and how I think we should be thinking about it. So the evidence regarding the merits of CHOP or CHOEP plus studies, for me, continues to be pretty disappointing. The vast diversity of these diseases, it's been hard to identify common pathogenetic themes, like what's the gene or biology we should go after. I think the epigenome between the data with hypomethylating agents and HDAC inhibitors is certainly rising to the top. All these single new agents, and you know them, duvelisib, the JAK6 inhibitor, EZH2, they're all identical. None of them are home runs, none of them. And in fact, the PI3 inhibitors are getting killed by the FDA these days. And so how do we leverage these suboptimal drugs? Because if we're all sitting at home waiting for Owen or Mubarak to generate that next 90% single agent drug, you may be sitting at home for a long time. And so I think we got to take the clues we have regarding these drugs that work and think about how we might leverage them to be better. Combinations, in my mind, are the way, and novel combinations trying to circumnavigate a lot of what we're doing with CHOP. And here's just a simple mathematical calculation. Here are all the classes of drugs I've identified as active: ADCs, hypomethylating, HDAC, EZH2, PRALA, PI3, checkpoint. All of these drugs by themselves are like 30% drugs. And if we start thinking about all the ways we combine doublets or triplets or quadruplets, rather than thinking about, what study can I do as a young investigator? Can I do something and add it to ICE and add this drug to CHOP and add this drug to GDP? I would say, sure, you can do that. But the opportunities here, I think, are much greater. And if we can understand these things biologically and look at the evidence for combining them, I think the combinatorial aspects with new drug combinations far exceeds anything you can envision with combination therapy. So I work with a remarkable team now down at the University of Virginia in Charlottesville, where Tom Loughran is the guy that discovered LGL at the Hutch. This is an amazing team with chemists and biologists, and my team funded extremely well from the NIH and American Cancer Society, FDA. We just got a $6 million grant to create an orphan blood cancer research program, which is now underway, and that's what's funding a lot of the new drugs that I'm telling you about. So I think the future is bright with collaborations like this, and if you've liked anything I've had to say, you can join us at the T-Cell Forum in beautiful La Jolla. We had the pleasure of having Mubarak there last year, where he gave a great presentation. And if you have an abstract or something to say, personally send me an email with what you'd like to talk about, and I will do my best to get you an opportunity to share that data with this international forum. So with that, thank you so much.